This protocol establishes a novel neuronal axon and astro-glia co-culture platform to analyze neuron-to-glia interactions using high-resolution imaging. First, assemble the microfluidic culture chamber. Prepare neuronal cultures and induce the neuronal axons to enter into the other side of the MCP. Next, add cultured astrocytes to establish a compartmentalized co-culture system. Results including time-lapse imaging with confocal immunofluorescence microscopy can help understand the interactions between axons, astroglia, and neuronal axons.
We first had the idea for this method when trying to separate the effect of neuronal process versus neuronal secretion to the GLT1 regulation. Typically, individuals new to the method will struggle with inducing axon into another part of the cellular compartment. It requires a high density of healthy neurons plated on the neuronal side with the neuron plated near the central channels.
The MCP open chambers are designed for compartmented cultures of different types of cells. Polyornithine and laminin coatings are necessary for plating neurons and astrocytes in the co-culture. First apply 1 mg per milliliter polyornithine solution to the sterile glass-bottomed dishes and incubate at 37 degrees Celsius overnight. Wash the coated glass-bottomed dishes 3 times with double distilled water and then place under a sterile fume hood to dry.